All right. Now I would like to invite our second presenter for today, Dr. Clement Wong from University Tunku Abdul Rahman, Malaysia. So Dr. Clement will be presenting his work on the use of microbial biocontrol consortia as a promising approach in managing Fusarium wilt of bananas. So please welcome Dr. Clement. Do we have?
Thank you, Dr. Aisha.
Yeah, thank you.
Yeah. Let me share my screen, yeah. Good afternoon, everyone. Can you hear me and see the slides?
Yes. Yeah, we can see your slide.
Okay, thank you. Let me minimize this. Okay, so good afternoon, everyone. My name is Dr. Clement. I'm from University Tunku Abdul Rahman, Malaysia. So I hope everyone is doing well during this COVID-19 pandemic. So today I'll be sharing part of my PhD work on using microbial biocontrol consortia as an approach to manage Fusarium wilt of bananas. So I'll bring you through some, these are some of the topics that I will share. So I'll brief you through our national banana production and its own nemesis, the Fusarium wilt. And I'll also talk about some of the biocontrol, one of the methods that is a biocontrol method to control Fusarium wilt. And I'll also be sharing some of our research findings. And last but not least, I'll talk about some of my thoughts on the challenges and future direction in the biocontrol of banana Fusarium wilt. You know, as Malaysians, we are very proud that we have various delicacies made from our local banana varieties. However, most of our banana varieties are susceptible to plant pathogens, such as the Fusarium wilt. And you know, if the Fusarium wilt is not controlled, most probably we'll lose these favorite delicacies of ours, you know, as our banana varieties are being destroyed by the Fusarium wilt. Okay, so I got this from our local newspaper in Malaysia. So to date, banana production ranked third after durian and pineapple. So even so, this clearly shows that Malaysians still very much like their bananas, okay, despite the recent surge in durian production as a result of export demand. So here's a chart to basically show you the wholesale and retail price of bananas. Between the year 2000 to 2018, the wholesale and retail price of bananas have almost tripled. Okay, most probably due to the infection of pests and diseases, and also higher production costs and consumer demand from this for this fruit. So you know, our friend here, our old friend here, the Panama wilt, has caused much destruction to our national banana production. One of the widespread causal agent is the Fusarium oxysporum, former specialist cubancy, and the tropical race 4 strain is very much widespread in Malaysia, and it has been currently renamed to Fusarium odoratissimum. And I think most of us here are very familiar with the symptoms of bananas infected with Fusarium wilt. You know, sick plants are actually characterized by their yellowing of all their leaves and also culm and root discoloration. Okay, so Mostert and her colleagues in 2017 actually carried out a study to understand the distribution of Fusarium wilt throughout Asia. So in Peninsular Malaysia alone, as you can see, most of the banana plantations are affected by the tropical race 4 strain. And we also have a diverse Fusarium wilt, which includes race 1, race 4, and also race 2. So as you can see here, these are tropical race 4 strain. Besides affecting cooking and dessert bananas, it is widespread throughout the Asian Pacific region, okay, and also some of the Middle Eastern countries, which include Israel, Sudan, Oman, or even Ethiopia. And recently, it has spread through Colombia and Peru in the year 2019 and 2021. So this calls for immediate action to prevent TR4 from spreading to other regions. So of course, most of us feel that developing resistant cultivars is the gold standard, is the ideal method to reduce disease spread. However, most of the breeding process can be time consuming. So there are also other methods of controlling Fusarium wilt or the TR4 strain. So some studies shows that the application of fungicides seem promising. However, we are anticipating more few results on how these fungicides actually, on the efficacy of these fungicides in controlling the tropical race 4. So therefore, we actually chose an alternative method, which is the biocontrol method, with these advantages in mind. One is that the microbes can be produced in a large quantity using less sophisticated equipments, and it can be mass produced. They can be mass produced easily into bioformulation, okay, as shared by Dr. Rosita. And most importantly, I think the biocontrol method is environmental friendly, since we are not using any chemical, since we are not using any since we are not using any chemical fungicides, so therefore we will have less issues of chemical residues in food. Okay, and of course, biocontrol agents has been found to improve crop growth as well as post-harvest quality of agricultural crops. Like most other methods, the biocontrol method has its own limitations. One is that we don't know whether pathogens could evolve resistance towards the biocontrol agents. Secondly, biocontrol method usually suppresses the pathogen population, but not total elimination. And of course, biocontrol efficacy can be affected by abiotic factors such as weather, your soil fertility, as well as the agricultural practices. So if you look at most of the research articles focusing on Fusarium wilt of banana, a huge part of disease management focuses on biological control method. To date, most of the biocontrol studies focus on the use of single strain. However, recently researchers has switched their gears towards using Microbial Consortia, simply because it is much more effective in reducing the disease severity. So this leads us to two research intentions here. Firstly is the use of multiple strain of BCA to control Fusarium wilt. As I mentioned previously, Microbial Consortia can actually reduce the disease severity, okay, better than the use of a single BCA, okay, most probably due to the multiple antagonisms exerted by the multiple BCAs. So we found that previous studies mostly focus on the use of multiple bacterial strains and fungal strains respectively. So far, there's no one that actually mix both fungal and bacterial strains together to test on their biocontrol efficacy against Fusarium wilt. So that is why we intend to mix both fungal and bacterial strains and see how they work. So the next part of the study is basically to formulate these microbial strains into formulations, into carriers. Okay, one reason is that we want to protect our microbes from environmental stress so that they can sustain their bioactivity as well as their viability while they do their job in protecting our banana plants from Fusarium wilt. So here is some of our findings. So we actually isolated potential bacterial and fungal biocontrol agents from healthy banana plants, and we subjected them to compatibility tests to see whether the bacteria and fungus are compatible with each other. And we found out that bacterial strain DRB1 and CBF2 has the lowest PIRG value. So the low PIRG value here is very important because at the end of the day, we do not want both bacteria competing against each other. It will not be beneficial for the control of Fusarium. So next, yeah. Okay, so after we have identified two of these biocontrol strains, so their molecular ID is Pseudomonas and Trichoderma. So we subjected them to detached leaf assay to see the ability whether they can reduce wilting severity. So from the table here, you can actually see that the application of both biocontrol agents were able to reduce wilting severity. So if you look at the cell death area, which is stained by trypan blue, you can see that leaves that are treated with BCAs, regardless whether it is single strain or double strain, the leaf cell death area has reduced significantly. And we have also looked at the potential of BCAs in protecting leaf mesophyll cells. So as you can see here, leaves that are treated with tropical race 4 alone showed a degradation of mesophyll cells, okay, so leading to the red ink being uniformly distributed throughout the leaves, okay, as compared to the leaves that are treated with biocontrol agents. Okay, so meaning to say biocontrol here plays an important role in protecting the leaf mesophyll cells from being degraded by fusaric acid, which is produced by Fusarium wilt during the course of invasion. So that is when we formulated 4 formulations here containing both biocontrol agents. So we have 4 carriers here. One is pasta granule, tech powder, alginate beads, as well as liquid formulation. So we applied these 4 individual formulations for a 3 months glasshouse trial, and we noticed that the pasta granules actually led to a lower leaf wilting severity, root, as well as comb discoloration compared to the other bioformulations, as well as the binomial fungicides application. So we actually proposed that the better disease controlling efficacy of pasta granules could be due to the carrier materials used. So pasta granules is actually consists of largely of semolina flour or even wheat flour. So we suspect that these microbes could have utilized them as a carbon source to survive in the soil and to exert better biocontrol efficacy as compared to the other formulations here. So visually, you can see that bananas that are treated with bioformulations have lesser comb and root discoloration as compared to the binomial application. So if you look visually, pasta granules actually has the least comb and root discoloration as compared to the other bioformulations here. So we also evaluate whether these bioformulations can improve the growth of bananas. So
Dr. Clement,
yes,
have 2 more minutes, yeah, to wrap up.
Okay, sure.
Yep.
So from here, you can notice that the bioformulations actually can improve the growth of bananas as compared to binomial, as well as as compared to the binomial and the control treatments. So we also need 180 days storage to see how well the microbials survive at room temperature as well as 4 degrees. So we see, if you notice that those bioformulations that are stored at 4 degrees has a better microbial viability. So specifically, actually showed a better viability of both Trichoderma and Pseudomonas strains up to 180 days. However, when you store pasta granules at room temperature, okay, both of the strains, the viability of both strains actually dropped to 0 after 90 days of storage at room temperature. So which means we have to do more work in improving the pasta granule formulation so that it can be stored at room temperature. So these are some of the possible future research directions. This will be the last slide. Give me a few more minutes. So of course, formulating a fertilizer is never easy because we have to know what are the microbes that we are choosing, whether they are producing spores or not, or the number of microbes that we are adding in, because this will lead to problems such as compatibility problems, and also whether they are able to survive in carrier materials. And of course, as I mentioned, we need to work more on improving the shelf life of bioformulations, because farmers, they don't have the luxury of refrigerating, keeping their fertilizers in refrigerators. And I think I think we also need to work on the antagonistic mechanisms of these microbial consortia. How do they actually exert their antagonistic properties beneath the soil? And as what Dr. Ganesan has presented earlier in the morning, utilizing biocontrol agents can help to improve post-harvest and sensory quality of banana fruits. So this is one area that we should look into, because I think most of the consumers nowadays, they do prefer bananas that look and taste good. And of course, last but not least, validating biocontrol efficacy is very for long-term is very essential, because we need to know how well these biocontrol agents work for prolonged periods of banana farming. So last but not least, I'd like to thank these beautiful people who have helped me along the way in this research. And I also thank the Secretariat for inviting me to give this talk. Thank you very much.
Okay, thank you very much, Dr. Clement Wong.
Thank you.
Very nice presentation. But in the interest of time, yeah, we could not give you questions.
Sure, no problem.
Yeah, but please look at the chat box. If you the participants would have questions for you, please entertain their questions. Yeah, in the chat box.
Okay.
Participants, you can just shoot your questions to Dr. Clement in the chat box. Thanks very much, Dr. Clement Wong.